Bismillahirrahmanirrahim. Today our topic is tissue culture. Tissue culture in plant is also called as hydroponics, while in animal tissue culture is called as cloning process. So here we are studying in the plant so called as tissue culture or hydroponics. Tissue culture was discovered by Thomas Bulwas in 1902. And the meaning of the term tissue culture is derived from two words. Tissue means the group of similar normal cells, while culture means growth substance. So definition is the formation of many similar or identical cells or tissues from a single cell or tissue in a culture medium is called tissue culture. Here the word is a growth substance culture or growth substance. So medium used for tissue culturing. And there are three types of medium which are used in the process of tissue culturing. One is broth culture medium, which is the liquid growth substance. Then here comes second one, agar culture medium, which is a semi-solid or gel growth substance. Then step culture medium, which is a solid growth substance. Then we have discussed earlier that the tissue culture for plant is called as hydroponics, while the tissue culture for animal is called as cloning process. The tissue culture for plants is called as hydroponics, while the tissue culturing for animal is called as cloning. And then the apparatus used in tissue culturing. First apparatus is cell culture hood or biosafety hood or laminar flow hood. These are the three names for a single apparatus, which makes the culture medium germs free. And then here is a sterilizer or autoclave, which is the second apparatus which makes the explant germ free. Explant from which we take the sample, and here is the germ free process of the culture medium, while sterilizer or autoclave makes the explant germ free. Then this third one is cell counter or cell cytometer. Hemacytometer. Cell means cell and counter means the machine which is used to counting the cells. So it shows the number of cells in a tissue or callus. And then here came water bath. Water bath provide water for tissue culturing. Then fifth apparatus is centrifuge, which is used as centrifuge machine separate the cells from tissues or callus, which we discussed in the last topic, that in a centrifugation we take the test tube, we put the test tube in the centrifuge machine and it starts revolving, which gives the cells on the basis of density. So here in this process we also use the centrifuge which separate the cell from tissue or callus. And then here came inverted microscope, the compound microscope with movable piston. It's called as inverted microscope. Then in this process we also use the refrigerator or freezer, which provide the -20 centigrade temperature providing machine is called as refrigerator or freezer, which gives -20 centigrade temperature. Then here comes the incubator, which gives humid CO2, and it is also a temperature regulating machine. Then here comes the cryo storage container, which provides the liquid nitrogen. The liquid nitrogen is used to preserve plants. The chemicals which preserve plant is liquid nitrogen, while the chemicals which preserve animal is called as formalin. We use so in plants we are discussing the hydroponic, that is the tissue culturing of plants, so we use the liquid nitrogen. When we discuss the cloning process, so we use the in cryo storage container we use the formalin. And the last one last apparatus we use is plant hormones, which are actually lipids. In plant hormone, plant contain three types of hormone. One is auxin, the second one is gibberellin, and the third one is cytokinin. Auxin promotes primary growth in plants, which is actually the lengthening of the plant, and gibberellin promotes secondary growth, which is actually the weakening of the plants, while cytokinin promotes cell division. Then here came steps involved in tissue culturing. First step is obtaining explant, and first step is obtaining explant, and the second one is make the explant germ free through sterilization. Through sterilization or autoclave, we make the explant germ free. Then here came the third step, that is preparation of the culture medium, and make the culture medium germ free with the help of the cell culture hood, which is the first apparatus we use in this process. Then the fifth point is put the explant in a culture medium, and it starts varying the size of explant, because the explant starts growth. That is, first it has a small size and then it becomes a very large size, starts growing and becomes very large, and this large cell is called as callus, which we have discussed earlier, but I explain here. Separate cells from the callus by centrifugation process, which we already discussed. Then there are few points comes that high cytokinin plus low oxygen make the callus cells into shoot formation. High cytokinin and low oxygen make the callus cell to form the shoot formation. Then low cytokinin and high oxygen make the callus cells to form the root formation. These are important MCQs. Then high cytokinin, high oxygen and high gibberellin converts the plantlet into plant. Plantlet means a short plant or immature plant, which convert the plantlet into the mature plant. Thank you. If you like my work, so please like, subscribe and share my channel with your friends.